Good morning everyone. In the previous class we had discussed regarding the inheritance of one gene, and also we had studied the two laws which were based upon the observations of the monohybrid cross, that is law of dominance and law of segregation. So in this today's class we will be discussing regarding incomplete dominance and codominance. So first let us see what is incomplete dominance. So incomplete dominance is a phenomenon where both the alleles of a character express incompletely, producing a new intermediate phenotype in the heterozygous condition is called incomplete dominance. So in the previous class we had already learned regarding the law of dominance. So what do you mean by dominance means obviously there are two alleles or two alternative forms of a gene. So when these alleles are in a heterozygous condition, heterozygous means when both the alleles are different. So in that condition, one allele which is going to express itself is called as a dominant one, and the one more allele which is not capable of expressing or which fails to express itself is said to be a recessive one. So in that case, out of two alleles, only one allele is going to express its character. So that is called as a complete dominance. So now coming to the incomplete dominance. So what is this incomplete dominance means? So when the experiments of monohybrid cross were repeated using the other traits in the other plants, means in the first case, that is in the monohybrid cross we had used the pea plant with a trait that is a stem height. But when these experiments were repeated using the different characters in the different plants or other traits in the other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. It means that it was not resembling a parent A or it was not resembling parent B. So, but the character was in between both the parent A and parent B. So, the inheritance, so the best example for this is the flower color in dog flower, or it is also called as a snapdragon, right? So, we'll see the explanation which is given by Corens. So when Corens crossed the homozygous red flowered plant with a homozygous white flowered plant, so surprisingly in F1 generation all the hybrids were pink flowered plants because the dominant gene R fails to mask the recessive gene R completely. So here Drager Mendel has taken the pea plant and he has selected the two traits, that is the homozygous tall and the homozygous dwarf. And then in the F1 generation he has observed that all the offsprings were of the heterozygous tall. But when Corens has done the same experiment with a different plant, that is a snapdragon, or it is also called as a dog flower. So using that, so he has taken one trait, that is the flower color. So one parent has a homozygous red flowered plant and one is the homozygous white flowered plant. So when he crossed these two in the F1 generation, usually according to Drager Mendel, he has to get all the plants to be red or either it has to be white. But here Corens did not get either red or white, but he got the character which is in between both these two, that is a combination of red and white, that is pink. So here what we can say is that, so the dominant gene is means it is not capable of completely masking or it is not able to suppress the other character. So this is nothing but the incomplete dominance. So when this F1 pink flowers were self-crossed, means again this F1 hybrids were self-crossed, and in the F2 generation what he got is that the 25% of the red color flowers, 50% of the pink flowers and 25% white color flowers. So with the ratio of 1:2:1. But when we have taken the Drager Mendel's expression, there we had got that the phenotype was in 3:1 and the genotype was 1:2:1, right? So here we are getting the phenotypic ratio as 1:2:1, means the phenotypic ratio in the monohybrid cross of pea plants was that is phenotypic ratio was 3:1, but here in the incomplete dominance the phenotypic ratio is 1:2:1. So we'll see with the schematic representation. So here you can see that in the parent generation we have taken two plants, so that is the two snapdragon plant. So the both the plants are going to differ in only one character, that is a flower color. So the one flower is of a red color and one more flower is of a white color, right? So here according to codominance, the red is a dominant one and white is a recessive one. So that's why we have marked the red color with the capital letters, and whereas the white is with a small letters. So when we take the gametes, that is a capital R and small r, in the F1 generation we are going to get a heterozygous flower, that is capital R and the small r. So this flower is having the color pink, so which is the character in between the parent A and parent B. So now when this F1 generation is self crossed, so what we are going to get is the phenotypic ratio and the genotypic ratio are both are same. Then when we take the phenotypic ratio, that will be 1:2:1, where one is red and one is pink, two are pink and one is white. So when we take the genotypic ratio, so that is like one is homozygous red, two are heterozygous pink and one is homozygous white. So but when we compare this to the monohybrid cross of the Reger Mendel, which he has taken the pea plant, there we had got the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. So here the phenotypic ratio is going to differ in the complete dominance and the incomplete dominance, right? So now let us study regarding the codominance. So here co means both. So both the alleles are dominant. So what it is going to state is that it is a phenomena of inheritance where both the alleles of heterozygous express themselves equally at a time. So here in the complete dominance, what we have seen that out of two alleles, only one parent is going to express. Means in the F1 generation, only one allele is going to express its character. Right? In the incomplete dominance, out of two parents or out of two alleles, the character is going to be in between these both the alleles. Whereas in the codominance, both the alleles are going to express the character. Right? So what is the best example for the codominance is human blood group. So that is, in the human, the blood group AB, means the persons who are having the blood group AB have the alleles IA and IB, which are expressing equally by producing both the sugars, that is antigen A and antigen B. So this is codominance, means both the alleles are going to express the character. So let us study regarding the human blood group. So ABO blood groups are controlled by the gene I. So every character is controlled by a gene, and here the ABO blood group is controlled by a gene I. But the thing is here, this gene is having 3 alleles. So every gene is usually having the 2 alternative forms, but in the case of ABO blood group, so the gene I is having 3 alleles. So that's why it is also one of the examples for the multiple alleles. Multiple alleles means, so the gene which is having more than 2 alleles is called as a multiple allele. So even the ABO blood group is having 3 alleles, that is IA, IB and a small I. So here the alleles IA and IB are going to produce a slightly different form of the sugar, while allele small I does not produce any sugar. So here capital IA and capital IB, so they are both are going to produce the sugar, whereas the small I, it is not going to produce any sugar. Because humans are the diploid organisms. So we are the diploid organisms. So that's why every person is going to possess only 2 out of 3 I gene alleles. Means the character or the blood group is, though it is controlled by 3 alleles, but every individual is going to have only 2 out of these 3 alleles, as we are the diploid organisms. So capital I and capital IB are completely dominant over the small I. In the other words, when IA and small I are present, only IA is going to express, and when IB and small I are present, IB is going to get expressed. So obviously IA and IB are completely dominant over the small I. So whenever the small I is present with the capital IA or capital IB, so only the IA and IB are going to be expressed, and they are going to suppress the small I. But when IA and IB are present together, then both express their own types of sugar. So this is because of a codominance. So as I said you, so what do you mean by codominance? Means together. So whichever alleles are present, they are going to completely express the character. So that is nothing but the codominance. So when this capital IA and capital IB are present together in an individual, so they both are going to express their character together, right? So complete dominance means only one character is expressed. Incomplete dominance means the character is in between the 2 alleles. Codominance means both the alleles are going to express the character. Or else we can say that in the complete dominance, F1 is going to resemble one of the parents. And in incomplete dominance, the F1 is going to resemble the character which is between the two parents. And in case of the codominance, F1 is going to resemble either of the two parents. Means both the parents are express the character in the F1 generation. So these are the three types. So then hence red blood cells have both A and B types of sugars. So now we can see here the table which is going to show the different genotypes which are present in the parents which are having a blood group A, B, AB and O. So since there are three different alleles, as I said you, capital IA, capital IB and small I. So usually there are 6 different combinations that are possible, and therefore we are going to have a total of 6 different genotypes of the human ABO blood group. So let them study them one by one. So here we can see the parent 1 and parent 2 means allele from parent 1 and allele from parent 2 in the second box. In the third column we can see the genotype of offspring, and in the fourth type we are going to see the blood types of the offspring. So if a parent is having a capital IA and capital IA, means both the parents are having capital IA, so then the genotype of offspring will be IA and IA, and the blood type is A blood group. So suppose if the parent 1 is having capital IA and parent 2 is having IB, so obviously the genotype are capital IA and capital IB. So then here both the alleles are going to express their character, and hence the blood group is AB. In the third one, if the parent 1 is having capital IA and if the parent 2 is having a small I, as I said you, this capital I is completely dominant over a small I. Here also the blood group will be A. So in the next one, if the parent 1 is having capital IB and if the parent B is having parent 2 is having capital IA, so then again both the genotypes or both the alleles are going to express their character, and again the blood group is AB. So suppose if the parent 1 is having allele IB and parent 2 is having allele IB, so here also the blood group is going to be B. So in the next case, if the parent 1 is having capital IB and the parent 2 is having a small I, so then the genotype of offspring will be capital IB and small I. So then the blood group will be B. So in the last case, that is if the parent 1 is having a small I and if the parent 2 is also having a allele I, so then the genotype of offspring will be small I, and here the blood group is going to be O. So here what we can say is that whenever there is a capital IA and capital IB, so you obviously they are going to express themselves. So if the capital I is present with a capital IA or small I, so it is going to be a blood group A. If the capital IB is going to be present with a capital IB or a small I, then it is going to be blood group B. If a small I is present with a small I, then the blood group will be O. So only in case of if it is having a IA and IB, so then the blood group is going to be AB. So there is and one more thing is that blood group O is also called as a universal donor, as it can donate the blood for a blood group A, blood group B or blood group AB. And blood group AB person is said to be a universal acceptor, as they can accept the blood from a blood group A, blood group B and blood group O. So now we will study the one more thing. So that comes before that we'll see what are multiple alleles. So more than 2 alternative forms of a single gene occupying same locus on the homologous chromosome and control a single character is called as a multiple alleles. So as I had said you before, if a gene is having more than 2 alleles, so then it is called as a multiple alleles, right? So in the blood group, so the best example is the ABO blood group in man. So the man is controlled by a gene I. So the man's blood group is controlled by a character, or the character or the blood group ABO blood group is controlled by a gene I, and it is having the 3 different forms of alleles, that is capital IA, capital IB and a small I. So here as the ABO blood group is having 3 alleles, so it is the best example for a multiple allele. Means more than 2 alternative forms of a single gene occupying same locus on the homologous chromosome and control a single character is called as multiple alleles. Right. So now we'll study the one more topic which comes under this. So that is a single gene product may produce more than one of it. So as I said that a single gene means a gene is going to control a character, right? So as it is having the capacity to produce the polypeptide chains or the protein, so it is going to control a character. So but a single gene product can also have more than one effect. So it's just what is the best example for that is, starch synthesis in pea seeds is controlled by one gene. It has two alleles. So obviously every gene is having two forms of alleles. Here also, that is, one is capital B and one is the small b, right? So this starch is synthesized effectively by the homozygotes, that is a capital B, and therefore large starch grains are produced. So the starch synthesis is controlled by two alleles, that is capital B and small b. So whenever the starch is synthesized effectively, so that will be in the homozygotes, that is a capital B and capital B. So obviously that grains will be large in size. So in contrast, means opposite to that, whenever the seeds are having the homozygotes, that is a small b and small b, so obviously they have lesser efficiency in starch synthesis and produce smaller starch grains, right? So whenever the starch synthesis is more, that means whenever the alleles, there are homozygotes, that is capital B and capital B, so obviously the synthesis of starch is more and obviously the grain will be larger in size. But whenever the homozygotes are having the recessive alleles, so that is a small b and small b, here there is not much effective synthesis of the starch. Hence the size of the grain will be smaller, right? So after the maturation of the seeds, capital B means the seeds with a capital B and capital B are round and the small b and small b seeds are wrinkled. So obviously the round seeds will have this large amount of the starch, whereas the wrinkled seeds they will be having a less amount of the starch. So the thing is that heterozygotes produce round seeds and so B seems to be dominant alleles. So whenever there is a heterozygous condition, means whenever the capital B is going to appear with a small b, obviously the seed is going to be round in shape. It means that the capital B is dominant over the small b. But the starch grains produced are of an intermediate size in the BB seeds, right? So here what we can see is that according to the monohybrid cross or the dominant law of dominance, so here according to that, if the capital B is dominant, then the F1 generation is going to have a round seeds, right? But here the seeds are round, though the shape is same, but the size is going to be intermediate, right? So here the one gene product is going to have more than one effect. It means that the gene is the gene is known for the starch synthesis, but it's also going to control the size, that is the size of a seed. So here the intermediate size is going to represent the one more effect of the gene. It means that a single gene product may produce more than one effect, right? So this is all for the today's class. So once again we'll just revise the things. So first we had studied the incomplete dominance. So when the Corans cross the means when he done the monohybrid cross in a snapdragon, so he saw that in the F1 generation, so the F1 is going to resemble, it is not going to resemble either of the parents. It is not going to resemble the A parent or the B parent. So but it is in between the two parents. So hence it is called as an incomplete dominance, and the phenotypic ratio is 1:2:1. So which is different from the monohybrid cross of the Reger-Mundell, right? So which is 3:1. So now after that we have studied the codominance, means where F1 is going to resemble both the parents, and which is the best example is the ABO blood group. So ABO blood group is also an example for a multiple alleles. So what do you mean by multiple alleles? Means if a gene is having more than two alleles, then it is called as a multiple alleles. Along with that, we had also seen that a single gene product can have more than one effect. Means a single gene product can have more than one effect, right? So that's all for the today's class. In the next class, we will be studying regarding the inheritance of two genes and linkage and recombination, and also the chromosomal theory of inheritance. Thank you.